all of these other structures in the eye, the goal of them is to help light focus right here. This is the fovea of the retina. The entire retina, though, contains photoreceptors that allow for phototransduction. So this is where I'm going to zoom into now, into the retina, and then we'll talk about how the fovea is important specifically in a bit. So light is coming in from this direction, as you can see here. And what we'll look at when we zoom into the retina is that the cells that actually do the phototransduction are located at the very back of the retina. Let's go ahead and look at this. So here is a zoom in picture of the retina. So we zoom in to this right here. This is what we see. This is the very back of the eye. So the choroid and sclera would be over here. And then the vitreous humor would be over here. And the light is going to actually pass through multiple layers of cells. They're primarily transparent, so light can pass through them, and it's going to hit the cells in the back. These cells back here, what do you think they are? Rods and cones. Those are photoreceptors. These are the cells that are able to change when light binds to them. So proteins are present within them that change shape when a photon of light binds, and they are able to transduce that light into a neural signal. So this is where visual information processing is initiated. There are two types of photoreceptors. So rods, these are very sensitive. They're able to detect low levels of light. So they work at nighttime. They are present in the periphery, opposed to in the fovea. Cones are located in the fovea where you're focusing. So when you're looking at me right now, the light is focused on your fovea. If you stare at me right now, you can see things in the periphery. I can see my hand over here, but I can't see it very well. If I go like this and I look at it, I can now see it very clearly. That's when your fovea is focused on something. Cones are responsible for color vision, cones color, and they have high numbers in the fovea. So you can see really well in color when you're focused on something. Cones also only work with high levels of light. So at nighttime, you actually can't see in color very well. If the lights are down low, you can't really see colors. You might know that an apple is red, but you can't actually tell it is if you don't have enough light. There are 3 types of cones I'll come back to as well that allow you to see different colors. So 3 types of cones, 1 type of rod. So that's what's happening back here. Of course, we're going to look at the detail of how that happens, how light becomes a neural signal. So it's going to change the membrane potential, and it's going to then communicate to the cells. The information is going to travel to bipolar cells. Bipolar cells are bipolar. Finally, we see some bipolar cells, meaning they have 2 processes coming off of a single cell body. Bipolar cells are going to transmit the information to ganglion cells. So these arrows going this way, this is neural information. Light goes this way, neural information goes that way. The axons of the ganglion cells become the optic nerve. So these axons are all going to travel out right here. What is this little region right here then? That's the optic disc. This is the location in the retina where there are no photoreceptors because the optic nerve is traveling through there. It has to pass through. This is a photograph of the retinal surface. You get one of these if you go to the eye doctor, where you can actually see a picture of the fovea. Is it on this dark spot right here? That's a high concentration of cones. And then the optic disc, that is what the axons of the ganglion cells that are becoming, that have become the optic nerve to carry this information to the brain. This is a location where there's no photoreceptors, so it's called a blind spot. This is your blind spot. You can find it in your periphery if you look for it.